Welcome to Let's Wellness and to our series on a fascinating world of the gut microbiome. I'm your host, Dr. Padma Ramurthy. In our last episode, we explored the uniqueness of each person's gut microbiome and how it evolves from birth to adulthood. Today, we are delving deeper into the composition of the gut microbiome and the factors that influence it. A person's gut microbiome is unique, much like a fingerprint. Your microbiome is different from your, say, brothers or your friends or your sisters, varying in diversity, meaning different species of bacteria. The more diverse species of good bacteria you have, the better it is for your health. Think of diversity as a buffet of healthy dishes versus a plain piece of toast for your health. Additionally, the number of each microorganism and the ratio between different species of bacteria also play a crucial role. Early life factors such as birth method, infant feeding, breastfeeding versus formula feeding, antibiotic use, all this shape your core microbiota. For instance, vaginal delivery and breastfeeding are associated with higher microbial diversity compared to C-section and formula feeding. In early development, the microbiota consists of fewer microbes, but diversity increases significantly during the first year of life. Bacteria such as Enterococcus, Clostridium, Streptococcus, they all play a role in immune system development. This initial microbial exposure influences the degree of immune response. Human milk enhances the production of IgA, immune cells like IgA, cytokines, and cytotoxic lymphocytes, creating a strong defense mechanism. Our gut microbiome is constantly changing over time and is influenced by numerous factors. The recent advances in molecular biology have made it possible to study these microbes in greater detail. As bacteria make up most of the flora in the colon and account for almost 60% of the fecal nitrogen, so how do we test for the bacteria in the gut microbiome? You guessed it right, the stools. The feces is an ideal source of gut flora for testing the gut microbiome. Scientists begin by extracting the nucleic acid from fecal specimens. They then utilize techniques such as bacterial 16S rRNA, gene sequencing, and whole genome testing to identify and categorize the bacteria in our gut. The gut microbiome test report provides an overview of the microorganisms in your gut and whether they are associated with various diseases and disorders. A few companies claim they can even tell you which foods are best for you to eat based on the composition of your microbiome and other personal data. Despite the uniqueness of each microbiome, certain core species are shared among most individuals. The two dominant phyla of bacteria, Firmicutes and Bacteroidetes, represent 90% of the gut microbiota. Research has identified links between certain good bacteria and markers of good health, as well as bad bacteria and markers of poor health. For instance, beneficial bacteria like Faecalibacterium and Bifidobacterium produce short-chain fatty acids that support gut health and modulate the immune system. More about short-chain fatty acids in our next episode. So the good bacteria enhance the production of short-chain fatty acids. On the other hand, harmful bacteria such as Clostridium perfringens and toxic strains of E. coli can trigger diseases and promote aging. Studies have shown that the gut microbiota's composition is influenced by geographical location, reflecting the impact of environmental factors. For example, a study found that Japanese individuals have unique gut bacteria capable of digesting seaweed, a staple in their diet. This highlights how our environment and diet can shape our gut microbiome. The microbial composition of the gut differs between individuals living in developed and developing countries. Epidemiological data show lower prevalence of allergic symptoms and asthma in developing countries, supporting the hygiene hypothesis. What's hygiene hypothesis? Excessive cleanliness in developed countries may reduce the infectious stimuli which is needed for the immune system development during childhood. Factors such as large families, rural living, and less antibiotic use in developing countries contribute to more diverse microbiota. The use of antibiotics also has a double-edged effect on the host organism. while they destroy the pathogenic microbes, they also disrupt beneficial ones, leading to less diverse microbiota. The overuse of antibiotics in agriculture, poultry, and cattle is another concern. Studies have shown that changes in gut microbiota due to overexposure to antibiotics can be linked to the development of chronic diseases such as asthma, food allergies, and autoimmune disorders. The rise of Western diet, rich in processed food and light on fresh fruits and vegetables, is taking its toll by pushing out the good bacteria and encouraging the bad bacteria. Artificial sweeteners derived from sources like paraffin and the petrol industry are particularly problematic as our bodies and microbes are not accustomed to breaking them down. Emulsifiers, common in processed foods, have been shown to reduce the microbial diversity and promote inflammatory microbes in animal studies. Gut microbes do things the gut can't do, we can't do. Liberating or synthesizing nutrients from food, especially from plants and their polyphenols, digesting the non-digestible substrates from fiber that we eat, and producing thousands of metabolites, useful chemicals that are involved with immunity and keeping our gut and colon healthy. To do all these functions, microbes, our friendly microbes, need their food. And what is their food? Fiber. Minimum of 30 grams of fiber a day. But the average intake in most countries where processed food has taken center stage is hardly 10 to 15 grams per day. So how can you help your gut microbes do their job more efficiently? Here are some tips. Eat a variety of plants. Aim for 30 or more different plants each week. A diverse diet supports a diverse microbiome. Include nuts, seeds, pulses, whole grains, fruits, and vegetables. Consume colorful foods. Berries and other colorful fruits are rich in polyphenols, which our microbes love. Polyphenols act as antioxidants and have anti-inflammatory properties. Include fermented foods in your diet. Kimchi, natural yogurt, kombucha are some of the examples of fermented foods. These fermented foods are rich in probiotics that can enhance your gut microbiome. Limit snacking. This can help maintain a balanced microbiome. Avoid ultra-processed foods. These products lack nutrients and fiber necessary for a healthy gut. Studies like the Twins UK and Predict have shown that even genetically identical individuals respond differently to the same foods, influenced by both microbiome and genetics. These studies highlight the importance of personalized approach to diet and nutrition. In conclusion, your gut microbiome is a unique and dynamic community influenced by numerous factors. By understanding the composition and influences of microbiome, we can take proactive steps to support its health. A diverse diet rich in plant-based foods, fermented products, and minimally processed foods can go a long way in maintaining a thriving gut microbiome. Stay tuned for our next episode where we'll dive deeper into each of these tips and how they can support a thriving gut microbiome. Thank you for joining me on this journey through the hidden world within us. Until next time, take care of your gut and it will take care of you. Thank you for tuning in. If you have any questions or topics you'd like me to cover, feel free to reach out. Don't forget to subscribe and share this podcast with anyone interested in gut health and wellness. See you in the next episode.